how do you treat a deadly disease when you literally don't even know what causes it? It's one of the most frustrating, honestly terrifying questions in modern medicine. Just imagine a clock ticking down, but the bomb squad can't even see the wires they're supposed to cut. Well, today, we're stepping into the shoes of medical detectives. We are going to explore exactly how researchers search for the biological mechanics behind a disease when its root cause is completely hidden in the shadows.
Welcome to This Explainer. Today, we're diving into the biological mechanics behind IPF, which is truly a data-driven medical mystery. Let's get right into it. Okay, let's dive into this. The disease we're focusing on is idiopathic pulmonary fibrosis, or IPF. Let's quickly break that down. Idiopathic, that literally means there's no known cause. Pulmonary means it relates to the lungs. And fibrosis, well, that means severe scarring. As this unexplained scarring progresses, it causes a critical loss of alveolar function, meaning those tiny air sacs in the lungs that exchange oxygen and carbon dioxide, they simply stop working properly.
To understand why finding the biological mechanics of this disease is just so incredibly urgent, you really only need to look at this time frame: 2.5 to 3.5 years. That is the median survival time after diagnosis for someone who learns they have IPF. The stakes here absolutely could not be higher. It's a rapidly moving disease, and because the initial cause is a mystery, doctors are constantly racing against this incredibly short biological clock.
And this brings us to an even tougher reality. The 5-year survival rate is only 20 to 40%. It's a highly serious disease with a poor prognosis, and right now, there is no cure. But here's exactly why uncovering those genetic clues through data is so vital. While we can't cure it yet, we do have treatments that can slow the damage and prolong life. Early detection is a real game changer here. If we can understand the mechanics of how this scarring happens at a microscopic level, we can intercept it much earlier. So how do we actually find those microscopic clues? Well, I want you to frame yourself as a researcher who's just been handed a massive bioinformatics dataset. Our main investigative tool for this mystery is bulk RNA sequencing, or RNA-seq. The researchers in the underlying study gathered 206 human lung tissue samples and used RNA-seq to quantify the messenger RNA content within them. We're officially zooming all the way in here. We're moving from the macro level of human lungs struggling to breathe, right down into the microscopic level of raw genetic data. To make sense of all this data, the study was split perfectly down the middle. We've got 103 tissue samples from patients with IPF compared directly against 103 control samples, meaning tissue without the disease. Now, what's really interesting about this methodology is that the researchers didn't just grab random samples. No way. These two cohorts were strictly frequency matched for age, sex, and race. Why does that matter? Well, think about it. If one group was significantly older, any genetic differences we found might just be due to age rather than the disease itself. By frequency matching them, we guarantee clean biological data, stripping away those confounding demographic factors. Let's look a little closer at where this data actually comes from. Starting with those 103 IPF samples, it's so important to humanize this data, you know? Every single data point represents a real person fighting this disease. 36 of these samples were surgical biopsies from living patients with confirmed IPF. And the remaining 67 samples, they came from whole lungs that were removed from IPF patients during transplant surgery. So those 67 samples represent the old, diseased lungs that were literally taken out to save the patient's life.
Now let's see how this builds our comparison group, the 103 control samples. Where exactly do you get healthy lung tissue? Well, 41 of these samples came from patients undergoing biopsies to check for lung cancer. The researchers took tissue from the histologically normal, non-cancerous parts of those lungs. The other 62 samples came from deceased organ donors who had absolutely no history of lung disease. There's a bit of dark irony in biological research, calling a deceased donor healthy, but for the sake of studying lung tissue, these samples were completely pristine. They make for perfect controls. And this brilliantly illustrates how we turn biological tissue into readable data. To decode these samples, the researchers followed a highly technical process. First, they extracted the messenger RNA, or mRNA, from all 206 tissue samples. Second, and to be super technically correct here, they didn't just count the mRNA strands directly, they created fragments of cDNA from that mRNA content. Third, and finally, they quantified how many of those fragments corresponded to specific genes. They measured the expression levels across a staggering 18,838 distinct genes. If you visualize the resulting dataset, you basically get a massive, unimaginably dense genetic spreadsheet. The layout features a row for every single one of those 18,838 distinct genes, and a column for every one of the 206 specific tissue samples, both the IPF and the controls.
Sitting right there in the cross-sections, the cell values represent the exact quantity of cDNA fragments found for that specific gene in that specific patient. The biological mechanics behind IPF are literally hiding somewhere inside the math of this giant spreadsheet. It's wild to think about.
To read this spreadsheet, we just need a really simple rule of translation for what we call gene expression. Think of it as a literal tally of how active each gene is in the lungs. A lower number in a cell means there was less mRNA content found. That means lower gene expression. Basically, that gene was quiet. A higher number, that means there was more mRNA content, which means higher gene expression. That gene was highly active, constantly working in that lung tissue. So, the crucial point is this: how do we actually use these tallies to find the disease? We use a process called differential expression analysis. This is the ultimate goal of our bioinformatics project. We are systematically comparing the data between our two conditions, the IPF lungs and the control lungs, to find genes that show significant differences in expression. We're looking for the genes that are wildly overactive in the scarred lungs or maybe completely shut down compared to the healthy ones. We are sifting through 18,838 genes to find the exact genetic needle in the haystack that's driving the biological mechanics behind this devastating fibrosis. Which brings us right to the edge of the actual discovery. We have the normal lungs. We have the diseased lungs. We've completely stripped away age, sex, and race. We've extracted the mRNA, built the giant matrix of 18,838 genes, and we have prepared our analysis tools. The table is set. So the question we walk away with today is this: what specific genetic clues are hiding inside these 18,838 rows of data? Which genes are secretly conspiring to scar the lungs of these patients? That is the true beauty of bioinformatics. The answer is already right there in the data, just waiting to be differentially expressed and discovered.